Hello, today we'll talk about fatty acid oxidation. Fatty acid oxidation occurs in three stages. First is the activation of fatty acids. That this step will occur in cytosol. Okay. Second step, it is transport. This activation is occurring in cytosol, so the transport of fatty acid will be into mitochondria. How it will transport, that we will discuss. And third and last is our beta oxidation, that is the major heading, oxidation. So here will be occurring the oxidation. So first is the activation of fatty acids. But firstly, how we are getting the fatty acids? You know what are the lipids? If we talk about, these are the triacyl glycerides. Triacyl glycerol, it degrades into diacyl glycerol and then monoacyl glycerol, okay, and then into glycerol. In these steps we are getting free fatty acids, okay, like this. So here we are getting, and which enzyme are used? That is lipase. We are the lipolysis is occurring, so this is lipase. Simple. Okay, now this glycerol will undergo certain other reaction, and free fatty acid will undergo fatty acid oxidation. For what? For energy purpose. As we know that fats are the storehouse of energy, and when we need energy, this is used. Now we'll see how much amount of energy is released. Firstly, it's activation. How the fatty acids are activated? So fatty acid firstly gets converted into, if we talk, this is acyl coenzyme A. Okay, there is one more step, but that is not important. We can directly convert fatty acid into acyl coenzyme A, but if you want to know, then fatty acid first converts to acyl adenylate, but no need. Okay, here what is happening? ATP is converted to, two phosphates are released, and this PPI, two phosphates are converting into two phosphates like this. Through what? This enzyme. This is pyrophosphatase. Halaki, it is not very important, but you should be knowing that if this is converted to this by pyrophosphatase, so this reaction becomes irreversible. Okay, and here you can see one ATP is converting into PPI and AMP. Normally what happens when ATP is broken, then it converts into ADP and phosphate. So we say that one ATP is used, but here what is happening? We are putting inside one ATP, but we are using two phosphates. So when we'll count how much of ATP we have used in activation, we will say 2 ATP are used in activation of the fatty acid. Okay, like this we use of 2 ATP for activation. Like this, it is activated. Now this acyl coenzyme A is activated. You know what is acyl? Acyl is nothing but just a chain, CH2 and then coenzyme A is there. This is acyl and this is coenzyme A. Okay, now this is activated. Activation happened in cytosol. Now this transport, we want this acyl coenzyme A to enter the mitochondria, but you know this mitochondria is good. Outer membrane is good, but inner membrane, it's very bad. It doesn't allow anyone to enter into his house. Okay, so what will happen? So there is a friend of this acyl coenzyme A who will help this acyl coenzyme A to enter in a disguise. How? We'll see here. In the inner mitochondrial membrane, okay, there is a protein, that is carnitine carrier protein. Okay, so this carnitine shuttle system, suppose this is inner mitochondrial membrane. Okay, so outside the inner mitochondrial membrane, this is CAT1 and on the inner side of the inner mitochondrial membrane there is an enzyme that is CAT2. Okay, what is CAT? C is for carnitine, C is for carnitine, A is for acyl, and T is for transferase. Why this kind of name? Wait, this carnitine acyl transferase. In books you will see instead of acyl you can also see palmitoyl. Okay, why palmitoyl? Because most frequently we use palmitic acid. So this R group, acyl group is general term, okay, so but palmitoyl is specific term. When we are using palmitic acid, then only we can use palmitoyl transferase, otherwise acyl. Okay, so anything you can write, you will get marks, no doubt. So this is carnitine acyl transferase, or if we are using palmitic acid, then carnitine palmitoyl transferase 1, and on the inner surface of the inner mitochondrial membrane we will use carnitine acyl transferase 2. It is already present. Now what will happen? Now this outside what was? Acyl coenzyme A. One was acyl coenzyme A, another was carnitine. Okay, the one who is this friend cat1, who is the friend of acyl coenzyme, what it will do? It will convert into acyl carnitine. This will happen. This is cat1 carnitine acyl transferase 1. This is inner mitochondrial membrane. Okay, this is inner mitochondrial membrane, this is the outer side, and this is the inner side of the inner mitochondrial membrane. So you can see here, this cat1 is what it is doing. This was acyl coenzyme A. This is the activated form of fatty acid which we produced. Okay, here what will happen? Coenzyme A will be separated from acyl group, and this carnitine, which is a carrier protein, what it will do is it will take the acyl group of the acyl coenzyme A, carnitine, and it form acyl carnitine, and then this acyl carnitine will go into the will enter the inner mitochondrial membrane, because inner mitochondrial membrane now thinks that this is a carnitine, and he hides acyl behind him because he is a good friend. So carnitine hides his friend behind him and go inside quietly. And then what will happen? There is another friend of them, that is cat2, because now the carnitine wants many of his friends to come inside, so carnitine wants to go outside again. So this acyl carnitine will get converted to carnitine through cat2. This is also carnitine acyl transferase 2. So it will get converted to carnitine, and inside there is an coenzyme A present already. Now this is very important. This coenzyme A, which was the part of acyl coenzyme A, now this and this coenzyme A, these two coenzyme A's are different. This is present inside and this is outside. This is lost over here only, and this coenzyme A then again binds with acyl and form acyl coenzyme A. Now acyl coenzyme A is inside the matrix, inside the mitochondria. This was the function of carnitine shuttle system, that he wants the acyl coenzyme A to enter with the help of this formation of acyl carnitine. Okay, this is clear. This is a transport. Now third, what was that? Third was beta oxidation proper. Then comes beta oxidation proper. How it happens? Let's see. Now this acyl coenzyme A and has entered the mitochondria. Okay. Now acyl coenzyme A. This is a series of reaction. So this I have written the beta oxidation steps, and there was a mnemonic of beta oxidation proper. O hot. O for oxidation, hydration, oxidation, and this is thiolase. Okay, what will happen now? First step, acyl coenzyme A. This is, I have made the acyl group is this, and this coenzyme A. Okay, what will happen? This coenzyme A. First step is oxidation. What is oxidation? Oxidation is either addition of oxygen or removal of hydrogen or electrons. Okay, so here what we'll do, will not add oxygen, but we will dehydrogenate. Okay, so here we will dehydrogenate. So what enzyme will be here? Dehydrogenase. What full name is acyl coenzyme A dehydrogenase. Okay, so what it will do, it will dehydrogenate it and make and oxidize it. So you can see here, from here HH is removed. Okay, when HH is removed, that is dehydrogenase and oxidation is occurring. When HH is removed, here will be double bond, and it will be enoyl coenzyme. En means double bond, and trans is transform enoyl coenzyme, and delta 2 is just the position of the double bond. It's first and second. Okay, so on the second position it's double bond, so it's enoyl coenzyme. Clear? Second is what? H means hydratase. Which hydratase will be there? Nothing, just the name of the substrate and then hydratase. Enoyl, this enoyl coenzyme A, hydratase. Now here will be, what will happen? Addition of water. How it addition of the water? Now this double bond will, like, now this thing, where it is, this double bond will go here, like on this bit. So double bond will come here will come negative, here will come positive, and on positive OH negative will be coming, and on the negative H will come, so CH will form CH2. Getting it? Let me tell you here, if I'm talking about it, this is chemistry which we have done earlier. What will happen here was double bond. Now due to this effect, I don't remember the name, but this effect of oxygen, it will move the oxygen on him. Oxygen is more electronegative, so this double bond will be over here. This is not necessary, but just for the explanation purpose, you need to know that where it will, OH is negative, so it will go on the positive carbon, and here negative will be coming, so H positive will be coming. So like this, the carbon near oxygen will get hydrogen, this H2, and the far away from oxygen will get OH negative form. Okay, this is the beta hydroxy acyl coenzyme. Why acyl coenzyme? It was already there. Now we have added the OH over here. This is same as it is, but this OH is added. Okay, so if we have added OH, so it is beta hydroxy acyl coenzyme. Why? Because this carbonyl carbon is first carbon, then afterwards alpha, beta. Okay, so this is beta hydroxy acyl coenzyme. Okay, now third one is oxidation again. Now what will happen here? Oxidation. How oxidation? By dehydrogenation. Here again dehydrogenation will occur, but with which? Same, beta hydroxy acyl coenzyme A dehydrogenase. Okay, this will happen. Okay, one more thing. When dehydrogenation is occurring, okay, like hydrogen, this enzyme is removing hydrogen from this molecule. Now who will get the hydrogen removed? Here FAD will get, and it will convert into FADH2, that is a reduced equivalent. Here what will happen? Here also dehydrogenase, beta hydroxy acyl coenzyme A dehydrogenase is working. Now who will take the hydrogen? Here NAD will take. Okay, now NAD plus will take into NADH plus H plus. Okay, so here NADH has taken. Okay, dehydrogenase. Now what will happen? Dehydrogenase, which dehydrogenase? This oxygen H, this is removed. This is removed, and here come the double bond. Now this double bond will form what? This is a keto group, no? This is a keto group. So it's on. Now this is first carbon, this is alpha, this is beta. So on beta carbon there is a keto group. So beta keto acyl coenzyme, as it is, it's there. Okay, beta keto acyl coenzyme. Then at last T is thiolase enzyme. Thiolase enzyme is the splitting enzyme. Now splitting will occur. How? From where? From here, alpha and beta. From beta carbon, the splitting will occur. That is why we call it beta oxidation. Why? Because from beta the chain will break. Okay, now this compound will remain here. This is acid. Acid means 2 carbon, acetyl coenzyme. Okay, this will be forming acetyl coenzyme, and the remaining R, CH2, this R whole form, this will remain as it is, and it will repeat the whole cycle till only acetyl coenzyme A, both sides are left. Getting my point? Suppose if there is palmitic acid. Palmitic acid is 16 carbon compound. Now how many cycles it will repeat to convert? Listen, 7 cycles. I'll tell you. 7 cycles. how many 2 into 7 is 14. Okay, 2 into 7 is 14, but we have 16 carbon, but we know that at last here also there will be acetyl coenzyme. So plus 2, 16. This I have done, that 2 carbon, acetyl group is 2 carbon, so 2 carbon is forming at the last, and 7 times the cycle is repeating, so 14. But at the end, not only 2, here also acetyl coenzyme A will be formed, so 2 plus 2, 4 at last. So means in 6 cycles 2 carbons are lost in the form of acetyl coenzyme A, but in last cycle 4 are lost. So you can say 12 plus 4, 16. Okay, like this the whole 16 carbon is lost. You can check another carbon chain also like this. Energetics and other aspects of this beta oxidation we'll discuss in next video. Thank you.